following is most commonly associated with a defect in nucleotide excision repair? A. Lynch syndrome. B. Xeroderma pigmentosum. C. Ataxia telangiectasia, Bloom syndrome. E. Fanconi anemia. Answer B. Xeroderma pigmentosum. Explanation: Xeroderma pigmentosum is caused by defects in nucleotide excision repair, leading to an inability to repair UV-induced pyrimidine dimers. This results in increased skin cancer risk, photosensitivity, and dry skin. Lynch syndrome, A, involves mismatch repair defects, and the other conditions listed are associated with different DNA repair mechanisms. 2. Which of the following repair mechanisms is primarily responsible for correcting mismatched nucleotides in newly synthesized DNA? A. Nucleotide excision repair. B. Homologous recombination. C. Base excision repair. D. Mismatch repair. E. Non-homologous end joining. Answer D. Mismatch repair. Explanation: mismatch repair is responsible for correcting mismatched nucleotides that are incorporated during DNA replication. It occurs predominantly in the S phase of the cell cycle. Nucleotide excision repair, A, corrects bulky lesions like pyrimidine dimers, and base excision repair, C, corrects single damaged bases. 3. A 10-year-old child with recurrent sunburns and hyperpigmentation of the skin presents with multiple skin cancers at an early age. Genetic testing reveals a defect in a DNA repair mechanism responsible for repairing bulky helix-distorting lesions. This defect is most likely in which repair pathway? A. Base excision repair. B. Mismatch repair. C. Nucleotide excision repair. D. Homologous recombination. E. Non-homologous end joining. Nucleotide excision repair. Explanation: the defect described in the question is most likely in nucleotide excision repair, which is responsible for repairing UV-induced pyrimidine dimers, a type of bulky helix-distorting lesion. This defect is characteristic of Xeroderma pigmentosum, which causes photosensitivity, skin cancer, and early onset of sunburns. 4. Which of the following is true about base excision repair? A. It occurs primarily during the G1 phase of the cell cycle. B. It repairs bulky helix-distorting lesions like pyrimidine dimers. C. It is crucial for repairing spontaneous or toxic deamination of bases. D. It occurs primarily during the M phase of the cell cycle. E. It requires 2 homologous DNA duplexes for repair. C. It is crucial for repairing spontaneous or toxic deamination of bases. Explanation: base excision repair is responsible for fixing small, non-helix distorting base lesions, such as those caused by spontaneous deamination of bases. The process involves base-specific glycosylase, AP endonuclease, and DNA polymerase. It occurs throughout the cell cycle, unlike nucleotide excision repair, which occurs mainly in G1. 5. A patient with a family history of colorectal cancer is diagnosed with Lynch syndrome. Which of the following is most likely to be defective in this patient's cells? A. Nucleotide excision repair. B. Mismatch repair. C. Base excision repair. D. Telomerase. E. Homologous recombination. Answer B. Mismatch repair. Explanation: Lynch syndrome, also known as hereditary non-polyposis colorectal cancer, HNPCC, is caused by defects in mismatch repair genes such as MSH2, MLH1, and others. This leads to an increased risk of colorectal and other cancers due to the accumulation of mutations in the genome during DNA replication. These questions cover key aspects of DNA repair mechanisms, including nucleotide excision repair,